Continue on the second part for immunological memory lecture. So we focus on immunization. Immunization is a type of artificial active immunity. So they are artificial, that means they are being introduced artificially, not naturally, and they are active. They are producing, they are stimulating protection by activating our own immune response, not by any drug or transfer of antibodies from another person. So immunization is a vaccinations against infectious diseases. Generally, we can say that vaccination is for microbes, infectious disease. But nowadays, scientists also developing, they are close to being successful in developing a vaccination to different type of disease such as cancers. I mean, they are going to be producing vaccine to protect us from certain type of cancers. So we have a look on the things that related to us. Immunization schedule in Malaysia. Everybody experience this. We will go through around 13 type of vaccines for various pathogens. We have BCG, hepatitis B. This is diphtheria, tetanus, pertussis, and then polio, and then hepatitis B, and Haemophilus influenza type B vaccine. So this is combination of more than 4 types of pathogen. And then measles in Sabah and MMR, JE in Sarawak, measles, rubella, diphtheria, tetanus. So this is the same thing. Diphtheria, tetanus. HPV. HPV is only for women, girls, and also tetanus. So do check out their brief information on all these vaccines, what they are, their definition, and what they are used for. These are very important to us. So vaccination was first discovered in 1929 by the British Dr. Edward Jenner. The history that Edward Jenner observed that milkmaids, I mean this is a woman that working to extract the milk from cows who had been infected by cowpox were protected. I mean these ladies infected by cowpox, a type of smallpox, but majority infected on cows. Maybe when they were working with cows, when they're getting the milking process, were protected from the more serious disease called smallpox. We have 2 types here. Smallpox, just for human, infecting human, the virus infecting human, and also a different version of smallpox that infecting cows, cowpox. So he observed that these ladies being infected by cowpox is very mild, not so serious the disease, and then they recovered from the disease. Once they recovered from the disease, if somehow they exposed to smallpox, they will not produce any disease. I mean smallpox disease, because they already protected earlier by virus from cowpox. Okay, Edward Jenner inoculated healthy people. I mean, he's introduced in the skin with cowpox and found that they were protected from smallpox. He take some of the pus and scrape the wound from cows, or from healthy people that recovered from cowpox, and he injected on the skin to another different people, and he found that people do not develop any smallpox. This form of immunization was called vaccination. So the name itself come from the word vaca, or cow. So vaccine. And then some scientists discovered that it is actually the protein code of the cowpox virus. I mean, the virus of cowpox is somehow similar to the virus of smallpox. So they are almost the same, but they are not producing the same serious disease of smallpox. That means smallpox virus is will produce more severe type of disease, smallpox, but cowpox not so much. But somehow the patient still being protected, because earlier the body already developed antibodies to cowpox, because the cowpox virus almost similar to the smallpox virus. Our immune system got confused, so the antibody can protect us both from the cowpox and also to the smallpox virus, like 2 in 1. The same virus, the same antibodies produced, effective to smallpox, also effective to effective to cowpox, and also effective against smallpox, because they are similar, almost similar. So our body produce antibodies that can react with both. The cowpox antigen stimulates the production of antibodies. These antibodies somehow effective against both viruses. So this is how scientists begin to discover a type of protection from infectious disease through vaccination. So now we look on types of vaccines. So there are many types of vaccines. Now our country is looking at vaccine from Sinovac Biotech, a China company, and also another vaccine from Pfizer by UK and US. Okay, types of I think they are developed somewhere in Singapore. I will check again. So the type of vaccines, we have whole microbe vaccines. I mean, the vaccine is being made from the whole pathogen. I mean, we are talking about COVID-19. A COVID-19 virus is being used, the whole virus. But of course, they should be killed, right? So the kill vaccines, how scientists kill the microbe, there are various ways, by using heat, formalin, etc. Example in SARS polio vaccine and typhoid. Second is the most effective type of vaccine, but the most dangerous one if not being prepared properly. The live vaccines, that means the microbe is still alive. So live vaccines, but they are attenuated, they are being weakened, strain. Not, they are the same species or close related species, but even though they are still alive, they will not produce the disease. So this organism cultured to reduce its pathogenicity. I mean, they reduce pathogenicity mean they will not produce the same disease as the pathogen one, but still retain some of the antigens of the virulent form. Example, Sabin polio vaccine and BCG. And we also have subunit vaccines. I mean, we are not using the whole microbe. We destroy and into small parts, to say, right? Use subunits instead of a whole organism. For example, toxoid is a vaccine produced against toxin produced by pathogens. So toxoid is by converting a toxin to a toxoid. I mean, toxin, very dangerous. They are converted to a toxoid, and so this toxoid is a reduced form of toxin. Still will induce production of antibodies, but the pharmacological activity of the toxin is reduced. Example, in tetanus toxin, diphtheria toxin, both the D for diphtheria and tetanus in DPT vaccine are toxoids. They mean they are actually toxin, but change to go through a treatment and they become toxoid, not so dangerous anymore. Second, polysaccharides. Use polysaccharides as antigen, usually linked to protein to get better stimulation of antibody production. So this polysaccharide are available in a large number of bacteria as a capsule. So example, strep, pneumonia, and the most popular one right now, the most popular one is mRNA, but they are the same, almost same category, the genetic material, either DNA, RNA or mRNA, messenger RNA being used. Example for COVID-19 vaccine. Most of the COVID-19 candidates, even the 2 or 3 candidates that going to be used by our country in the near future is being made from mRNA. Okay, I will brief a bit on issue on this mRNA. I'm not talking that COVID-19 vaccine that going to be introduced to us is dangerous. We never know, but before vaccine for COVID-19, all those candidates from China, from UK, there is no mRNA vaccine has been used to human. This will be the first vaccine made from mRNA. I mean, the vaccine that they are talking right now, the vaccine for COVID-19, made from mRNA. So this is the first time that we scientists or doctors will apply mRNA as a vaccine for COVID-19. Why? Because previously scientists did try to develop a lot of vaccine from mRNA, but most of the time they found that the mRNA vaccine producing so many serious side effects. So there are a lot of side effects. So you can Google this, you can look on Wikipedia. So that's why none have been adapted to be used officially on human. So this time around, they will begin the first time in history officially using mRNA as a vaccine. Okay, do check this one out, because yeah, maybe all of us have higher chances of getting this in a very near future. So this is some sort of things that you need to know. Okay, we look on just to see to imagine what actually happening in modification of toxin to toxoid. So we can look here, toxin. They have all the needed component to make sure that they have the ability to become toxin. So they have this toxin moiety here, for example. Somehow, or scientists will bring them to go through a few things. The toxin moiety is not there anymore, so they are still the same, 90% or 99% the same molecule, the same toxin, but because a part of it that making it so dangerous already being taken off, so now they become toxoid, so they are harmless or not so dangerous anymore, but they still have the antigenic determinants. So since they already they still retain their antigenic determinant, they still can stimulate our adaptive immune response to produce antibodies. Okay, these are the references. Alright guys,